Hey everyone, we'll start on top of the hour, and in the meantime, feel free to check out the in the chat the lab website and also the article our guest speaker will be discussing, and on top is pinned the PDF file for the slides that Dr. Talbot will use for the presentation. And feel free to share the room if you think this is really interesting. I think it's really interesting to have this collaboration of or neurons also being able to perform immune surveillance. I think that's really interesting and a new field. So will be a really interesting talk. So yeah, thanks for coming. Hi.
Hello. I'm losing my voice today. How's it going?
Okay. Not sure if the app is working. Okay. Hey everyone. We will start shortly, and in the meantime, feel free to read the paper that I shared in the chat and also the lab website. Our guest speaker will be here. He's texting me. So let me give the link here.
Hi.
Hey. Oh, there you are. Perfect, Sebastian. How are you?
Do you hear me?
Yes, yes.
Oh, fantastic. Sorry about that.
Oh, don't worry. I've posted the link on top, so you should be everyone should be able to access it. And thank you so much for coming. That's perfect. It's right on time. So,
Perfect.
yeah, if everyone is ready, we can slowly start. I know people will still be even coming in, so but we can start with introduction and so on. So, um, so welcome everyone to Science Society, and a special welcome to you, Sebastian. Before we start, I'll give the audience a short introduction so that they learn a little bit about you, and then we go from there. So, and let me, I'm sorry, let me really quick share on Twitter that you were here. And, okay, perfect. Sorry about that. Okay, so Dr. Sebastian Talbot, he is a tenured associate professor at the Department of Biomedical and Molecular Sciences at Queen's University. And he did his bachelor in pharmacology at the University of Sherbrooke in Canada, and later on his PhD in physiology at the University of Montreal, also in Canada. And then he did an internship in Brazil, actually, at the Pontifical Catholic University of Rio. And then he did his postdoc at Boston Children's Hospital in the US. And he won many awards and fellowships, really a lot. And so Young Investigator Award, the most recent one was the Effervescence Star Award from Gilead Sciences and Montreal in Vivo in 2021. Yeah, before we start with the presentation, we usually ask like a couple of interview questions. So first of all, again, welcome. Thank you for coming. And if you could like let us know what motivated you or brought you to science, like to choose like a life in science. Was it something you maybe always wanted to do, or I don't know, a class or a book or whatever the story is? Thank you so much.
Sure. Thank you. Thank you, Katarina, for the invitation. Good afternoon or good night to everyone online. This is great to be here. Yeah. So, what brought me to science? Good question. I guess I had a friend that was slightly older than me, a neighbor, and my family bought us some of these chemistry set like that you do at home, right, with the little microscope and whatnot. And we built our little lab in my basement, and from there we started to do very basic experimentation, which really I thought was quite exciting, using the microscope and seeing what's happening, you know, in basically whatever on leaf or on the grass or in the garden. And that really I thought it was quite exciting, and that really sparked my curiosity to do science. And from there, I actually never looked back. So from when I was, I don't know, 10, 11 years old, all the way to now, I never actually looked back, and I just continued to be excited about studying new things. And yeah, that brought me here now.
Well, that's really interesting. It was very similar for me. Like my father, he was a physics and chemistry teacher
Hmm.
in high school, and he brought me home also microscopes and chemistry sets. The only difference is that I always just put things together that were written on never mix dangerous. At some point, my father took it away because it was getting too dangerous. Because everything else was just written what will happen. So it was
Yeah,
too boring. I wanted to know what will happen. Why did they say it's dangerous?
yeah. No, I get that. I get that.
um,
Actually, the same thing happened. We had those camcorder, right, to do like video back in the days, the big one, right? And my father actually found some of our recording of our experiment. It was like, no, no, you need to stop this because you're gonna set the house on fire. So that was yeah. So we needed to change a little bit the kind of experiment we're doing. So quite similar, I guess.
well, it's the curiosity of just figuring out what people don't tell us, what they already know. I guess. I don't know. Maybe that's it. Um, yeah, and then from there, how did you come about to do this really interesting and novel research project? If you could take us there, maybe there's an interesting background story. Sometimes, exciting projects like this, people didn't believe you, or did they believe you? Was it easy to get funding? Yeah, if you could share a
Sure.
little bit of a background.
So back in the PhD, I was doing neuroimmunology and studying basically diabetic pain. And so it was spinal cord and microglia. Then I moved to Boston to do my postdoc, where I started to do neuroimmunology, but in periphery, where we used to look at now nociceptor control, airway inflammation. So it was in the context of asthma. And so we published our first paper back in 2015, and kind of to started my own line of research. Eventually, the idea was to move to be to have my own lab. So I saw a paper in Science, and they publish that every year, basically the discovery of the year, right, in Science. And one of them was about immune checkpoint blocker and how it was changing cancer treatment. And then I asked my PI if I could look at whether neuron play a role in this, and then that's what started the project. So we started in 2014. It took quite a long time to get everything together for the publication. Um, and I, I I think people were actually excited about it from the start. So it was quite hard to publish, but to get the funding, and I think people were supportive of the idea, right? So I don't I think it was not that controversial, right? And so that was okay. It was just a long journey to get it published. Um, and yeah, basically, so that was the Science Breakthrough Paper of the Year 2013. And actually an investigator at Albert Einstein University, Paul Frenette, which recently passed, unfortunately, had really cool paper in Science where it was looking at the role of adrenergic neuron, so not sensory neuron, but adrenergic neuron in prostate cancer. And so putting that discovery of the year paper and the work of Paul Frenette, we thought that it could be a cool thing to look at sensory neuron in cancer.
Yeah, that's really interesting, and it's really exciting. I don't know if you read, but in the recent Nature a journal, there was an article about that, the really groundbreaking publications and research is declining constantly.
I saw that.
Um,
Yes, I really, yeah.
yeah, so congratulations. And what do you think? if that's okay, if I can ask you ahead of time, what
Oh
do
yeah,
you
yeah.
think is the problem? Because you don't have the problem, but you know.
Well, no, no, I saw the research. I read the paper. Um, I guess what the paper is arguing that whatever research is published now, whichever journal is less impactful or less disruptive, right? So basically, what it says is like even if you publish in um, let's say high impact journal, it's just the consequences is less impactful for the society. And what I don't know why it would be like that, or why the research in general is at less or produce less changes in the society, or less drugs, or less I'm not quite sure what's the reason for that. Maybe I don't know. Maybe the main, I don't want to say that, but maybe some of the key mechanism have been unraveled, and people have created drug to target them. So maybe that's one of the things. Maybe the low hanging fruit, or which the discovery that produced the most impact, and maybe the easiest has been found, and maybe that's why the consequences of this research has produced more um, impact in the society. And how we move from here to there to produce more impact for research, that's a great question. Unfortunately, I don't really have a great insight on that, really. We know that's more and more difficult to get drugs accepted by the FDAs. So yeah,
Yeah,
that's a great question. Actually, I don't know how we
I
move.
mean, my
How we move from
personal
there.
experience is that the things I really would like to do, I didn't get funding for. And
Right,
the things that are kind of, okay, it's the obvious next step to do.
right.
Those, get funded. And I think that's also a big reason,
Yeah, that you
maybe.
no. Yeah, you're definitely.
And then the
Yeah,
pharma companies are not pretty much the same, probably like low risk. But I feel like actually from Asian countries, from China, there's a lot of stuff coming out that is kind of surprising and pretty cool. And but anyways, it's a very long discussion, I guess.
no, I mean it's a cool discussion to have, though, and I think you're probably right. Where funding is more l- lower risk, lower reward, probably is one of the reason for that, right? So
Yeah, and I
yeah.
don't feel like that the NIH program officers are the problem. When you write them, they say, "Oh my God, this is amazing! You should apply for this high risk grant," and then it gets totally taken apart by the like by other researchers that say, "Oh, who are you to say this?" Basically,
Yeah,
I
yeah,
don't
yeah,
know. It's not necessarily the NIH people, it's more us. I don't
yeah.
know.
We're
I think.
we're we're tearing each other up. Yeah, probably that's true.
Yeah,
Yeah, yeah,
maybe.
yeah,
I
yeah,
don't know.
yeah, yeah.
But, anyways, this was a great pre discussion. Thank you for that. And now the stage is yours. The presentation link is on top of the room. So, please, everyone, feel free to access it. And yeah, thank you so much.
Thank you. So, should I open my camera, or is it just chat,
Oh,
or is
yeah,
there?
it's just everyone will open this by themselves. So, if you could
But
just say when you switch slides, to tell people
okay,
to
but
switch. Yep.
so it's mostly audio, right? There's not like
Yeah,
video.
exactly. No video.
Okay, okay, cool. Yeah, sounds good. Okay, yeah, and let me click on the slide. Yeah. So yeah, and guys, feel free to interrupt anytime. I think the idea is just to chat about the work, right? So if there's anything unclear, feel free to interrupt me. I think I have about 30 minutes of slides. So thanks everyone to be here. Thanks Katerina to invite me. So today I'm going to briefly introduce our work on nociceptor neuron effect on cancer immunosurveillance. So if we pass on the slide number 2, I'm going to briefly introduce what are immune checkpoint receptor. So to activate the immune system against danger antigen presenting cell, provide and key co-stimulatory signal to activate T cell to mount immune responses against bacteria and viruses. When those immune responses needs, when the inflammation needs to be resolved, so when we need to stop the activation of the immune system against, let's say, those bacteria and viruses, the same antigen presenting cell provide co-inhibitory signal to stop the activation of the T cell. This is really important. Otherwise, those T cell could probably lead to they would stay activated, and that could probably lead to autoimmune responses. So the way those antigen presenting cells provide those inhibitory signals is they use actually a ligand present on the surface of their of the APCs, and those ligands are called immune checkpoint ligand, and they activate receptor on the T cell, which are called immune checkpoint receptors. So the ligands bind on the receptor of the T cell, and that stop the activation of the T cell. If we pass on slide number 3, the consequences of the activation of those immune checkpoint receptor is T cell exhaustion. What does that mean is that the T cell will eventually lead or lose a capacity to produce cytotoxic molecule. They will lose the capacity to proliferate. They will lose the capacity to produce IL-2. So first, lose capacity to proliferate, lose the capacity to produce IL-2, and eventually they lose capacity to produce cytotoxic molecules. So the T cell will actually begin to be what we call quiescent. So they will, it's not really apoptosis of the T cell, but they will be there, but not activated. If we pass on slide number 4, and this is what we actually chat during the introduction, is that what people realize is that tumor cells are able to take advantage of this mechanism. So basically, tumor-specific T cell, upon the constant pressure of the immune system, will start expressing immune checkpoint ligands. So the tumor cells receive the pressure of the immune system that eventually want to kill them, and so the tumor cell want to adapt, so they start expressing those immune checkpoint ligands. And the immune checkpoint ligands will bind on the receptor of the T cell and stop their activation. So what oncologists realize is that if you block either the receptor or the ligand using specific antibody, you're able to prevent the exhaustion of the T cell, and you're able to safeguard the activity of the T cell against the tumor. And that really lead to to smaller tumor, right? And that really increases survival of patients. And that really transformed the treatment of many cancer, including melanoma. And this is what we see in the graphic on the right. So if we pass on slide number 5, so there's not only the immune system that help to protect the organism, there's also the nervous system, right? And when you looked at both systems, both the nervous and the immune system, you realize that they function in a quite similar manner. They will detect danger, integrate and process the information, generate a memory of the insult. They will produce effector responses that can be local or systemic that will target the resolution of the inflammation. So both the immune and nervous system, they function quite the same way, and they actually want to detect danger and protect the homeostasis. And when you looked at both systems, they have their pros and their cons. For instance, the immune system, slide number 6. So the immune system is highly mobile and highly specialized. However, it has a slow response time, while the nervous system allows for a very quick transfer of the information. However, it has a fixed organization and it's much less diverse. So from this, we realize that the immune system and nervous system are actually highly complementary. So then on slide number 7, I'm going to briefly introduce what are nociceptors. So nociceptors, they are a subtype of sensory neuron that are specialized in detecting environmental danger. They do that by expressing specific ion channel receptor on their surface that will detect these different types of danger. If we looked on the sensory nervous system here, there's a molecule called TRPV1 that is specialized in detecting noxious heat. So when there's noxious heat, you activate TRPV1, TRPV1 being an ion channel that will lead to the opening of the ion channel, and that will lead to the influx of cation inside of the sensory neuron. And that will lead to the generation of an action potential that travel from the periphery to the brain, and that generate an avoidance responses, which we call the withdrawal responses. So basically, you activate your sensory neuron, there's a feedback loop inside the spinal cord that will lead to withdrawal reflexes. There's also electrical signal that goes to the brain, and there's the perception of pain. So sensory neurons detect those danger, as I mentioned, using diverse ion channel receptor. The key one are TRPV1, TRPA1, TRPM8, and piezo2. TRPV1 being the detection of heat, TRPM8 the detection of cold, and piezo2 mechanical pressure or noxious mechanical pressure. If we go on slide number 8, this is about neurogenic inflammation. So what happened is when you activate, as I mentioned, those ion channel receptor, you generate action potential that travel from the periphery to the brain. There's the perception of pain, but those electrical signal, they can also travel antidromically, so back to the periphery, and that will lead to the local release of neuropeptides. And these neuropeptides have diverse action. If we look on the right of the slide, you will see that these neuropeptides will act on capillaries. They will increase the blood flow of these capillaries. They will increase the vasodilatation in these capillaries. That will also lead to or increase the chemotaxis of immune cells. Eventually, those neuropeptides will loosen the tight junction of the capillaries, and that will lead to the local, the extravasation of these immune cells. And that really put immune cells in very close vicinity with sensory neurons. And that will allow the crosstalk between the sensory neuron and the immune cell. So there's really a local crosstalk between the two, and this is due to neurogenic inflammation. So if we pass on slide number 9, neurogenic inflammation, it has been studied in various context, one of which, as I mentioned during my postdoc, we're looking at it in asthma. And one of the question is, or one of the unanswered question is neurogenic inflammation can also play a role during cancer? And we thought it would be an interesting question because it has been appreciated for a very long time that actually tumors are innervated, but we're not quite aware of the role of this innervation. So here you see a JXMed paper basically from 1897 that shows that a skin carcinoma is innervated by different types of neuron. We don't know exactly what types of neuron because the paper is very old, but so it's been very long appreciated that tumor are innervated. We're unaware of the role of this innervation, and we thought that via neurogenic inflammation, maybe the neuron could crosstalk with the immune system, and that could be an interesting question. So if we look at slide number 10, this is basically a cancer research review that shows that neurons, not necessarily sensory neuron, but neuron in general, could have potentially diverse role in cancer, right? It has been hypothesized that neuron could directly control cancer cell growth. They could potentially control neoangiogenesis. They were hypothesized that they would control inflammation, so directly act on the immune system. And there's some paper actually show that cancer cell can use a neuron to metastasize away from the primary site. So basically, neuron locally could have diverse function in cancer, and we choose to study whether neuron could crosstalk with the immune cells. So we decided to study this in the context of melanoma, so skin cancer. We choose skin cancer because, first of all, the skin is very densely innervated by sensory neuron. And I'm not a cancer researcher, so I thought it would be easier for us to actually use this model, as we can directly just inject cancer cell line inside or intradermally in the mice, and we can fairly easily monitor the growth of the cancer cell. So what we did is we use a very aggressive type of skin cancer, which is called B16F10. We injected to littermate control as well as nociceptor neuron ablated mice. We let the tumor grow for about 15 to 20 days. We looked at the presence of itch or pain in the mouse. After 15 to 20 days, what we do is we harvest the tumor. We use flow cytometry to look at immune cell exhaustion, and we did immunohistology to look at whether there's the presence of nerve inside of the tumor. So if we looked on slide number 12, this is our working hypothesis. We had 3 aims, and this is actually how I built my first grant. So first of all, we ask ourselves, are cancer innervated by nociceptor? Second, we ask ourselves, if those nociceptors are present inside of the cancer, could they locally release neuropeptide? One of the neuropeptide we choose to study is called CGRP. So whether this locally produced CGRP could act on T cell and increase immune checkpoint receptor expression of T cell, and if that would be true, that will lead to the exhaustion of the T cell. And the third question we ask ourselves is, if we ablate those nociceptor neuron, or if we block their function pharmacologically, are we able to prevent immune cell exhaustion and therefore have a smaller tumor growth? So this is our basically our 3 aim. I'm going to briefly touch on each of these aims. So first question, if we go on slide number 13, are cancer innervated? So if we go on slide 14, we first use a melanoma biopsy from patients, and then we stain those biopsy using H&E as well as TRPV1. And we basically what we found is, in comparison to adjacent intact tissue, the tumor themselves showed higher presence of TRPV1 being a marker of these nociceptor neurons. And then basically what we did is we used the clinical chart from the patient, and we tried to correlate whether the presence of these TRPV1 neurons, or the increased presence of these TRPV1 neurons, could correlate with some clinical feature observed in the patient. And what we found is that it's pretty small here, but I think it's like a 0.6 correlation between the presence of TRPV1 inside the tumor and the increased presence of immune cells inside of the tumor. So basically what slide 14 showed is that tumors from patients are innervated by TRPV1 neurons. Then if we go on slide 15, we ask ourselves, is it also the case in the mouse? So what we did is we use our cancer cell line, so B16F10, in this case it's EGFP, so it's labeled in green, and we injected this tumor in the flank of a mouse, the flank of an NEV1.8 critical tomato mouse, so a mouse that basically has all the nociceptor being labeled. In this case, is like a pink, magenta kind of color. And what we found is, after 22 days and using iDisco, is that there's a presence of nociceptor neuron in and around the melanoma. So on slide 16, we ask ourselves, if there's the presence of the nerve inside of the tumor, are these nerves actually more sensitive, right? Is the cancer cell able to sensitize sensory neuron and cause pain? And to test this, we decided to use calcium microscopy. And so what we did is we harvest nociceptor neuron from the mouse, we culture them in the presence or absence of the cancer cell, and then we load this co-culture with the calcium responsive dye, which is called Fura2AM. And what we do is we're able to flow different substances on the surface of the neuron and see if they respond more or less of these different substances. And so we use 3 different substances, which are, classic noxious stimuli: ATP, which binds to P2X2 receptor, AITC, which binds to TRPA1, and capsaicin, which binds to TRPV1. And what we did is we use really low level of these noxious stimuli, right? And what we found is that in neuron alone, in the blue, we found that there's very few neurons that respond to those noxious stimuli. However, when the neurons were co-cultured with a cancer cell, we found that they show higher responsiveness to these different noxious stimuli, which would suggest that the presence of cancer cell next to the neuron actually sensitize their response to these various noxious ligands. So if we go on the right of the graph, we ask ourselves whether the presence or absence of cancer cell would sensitize the receptor neuron, but in the mouse. So here what we did is we injected the cancer cell in the left hind paw of the mouse. We let the cancer grow for about 14 days, and then we harvest L3 to L5 DRG neurons, so the neuron that actually innervate the hind paw. We put them in a dish again, and we did calcium microscopy. And in comparison to the mouse that received keratinocytes, which are non-tumorigenic, we found that the mice that received the cancer cell their neuron actually show higher responsiveness to ATP and capsaicin. So both in vivo and in vitro, the presence of cancer cell actually sensitizes the receptor neuron response to noxious ligands. So if we go on slide 17, we ask ourselves, how is this happening, right? How cancer cells are sensitizing the response of nociceptor neuron? So to do this, we did a pretty complex triple co-culture experiment where we cultured neuron alone, CD8 T cell alone, or cancer cell alone, and then we co-cultured them with one another. And then we fax purified these cells, and then we did RNA sequencing. And what we found, if you look at the graph on the right, what we found is when cancer cells are co-cultured with neuron and CD8 T cell, they actually overexpress a molecule called SLIPy. And so we thought that was quite interesting because some of our work from postdoc, which was unpublished, actually found that SLIPy was a molecule that was inducing pain. So we decided to really look at whether SLIPy, produced by cancer cell, were able to activate sensory neurons. So if we go on slide 18, first of all, we wanted to see, well, is SLIPy actually released by the cancer cell? So then again, we co-cultured the cancer cell in the presence or absence of a CD8 T cell neuron. And we found that when the cancer cell are co-cultured with the neuron and the CD8 T cell, there's increased release of SLIPy. Then in the middle panel, we tested whether SLIPy can directly sensitize the receptor neuron. Again, we did a calcium microscopy. And what we found is when you expose cultured DRG neuron to SLIPy, it was able to induce calcium flux in these neurons. And we found that these neurons that respond to SLIPy were also capsaicin and KCl responsive, which shows or support the idea that these neurons are actually sensory neurons. So if you go on slide 19, we actually bin the SLIPy responsive neuron by size, and we found that the SLIPy responsive neuron were mostly small to medium size, and as I mentioned, they were mostly capsaicin responsive. That's what we found in the Venn diagram, again supporting the idea that SLIPy activate nociceptor neurons. Then finally, we ask ourselves, well, is SLIPy directly able to activate nociceptor neuron? Is SLIPy able to lead to the release of neuropeptide by these neurons? So to do this, we culture the DRG neuron, expose them to SLIPy, and we measure CGRP release. And we found that yes, SLIPy is able to induce the release of the neuropeptide CGRP. And finally, on the right-hand side of the slide, we ask ourselves, is SLIPy, when injected in vivo, able to induce thermal nociceptive pain? And we found that yes, it is the case. When you inject it in the left hind part of the mice, it induced transient thermal hypersensitivity in the mouse. So if we go on slide 20, so far basically what we found is that cancer cell release SLIPy, activate nociceptor neuron in response are able to release neuropeptide. And then we ask ourselves, well, is the presence of melanoma, not just SLIPy, is the presence of melanoma able to or increase the release of neuropeptide from these nociceptors? So again, co-culture the neuron in the presence or absence of the cancer cell. And if you looked on the right-hand side of the slide, we found that when the neuron in blue, they release a little bit of CGRP, but when they are co-cultured with the cancer cell in red, we see that there's increased CGRP release from these neurons. So then when we go on slide 21, we finally ask ourselves, is the presence of the neuron not only is the presence of the cancer cell not only sensitize the nociceptor, but does it actually modify their transcriptomic profile? And so to do this, again, co-culture neuron in the presence or absence of the cancer cell, fax purified them, and we did RNA sequencing. And what we found is that when the neuron are co-cultured with a cancer cell, there's increased calci expression inside the neuron, calci being the gene that encodes for CGRP. So if we go on slide 22, I can just give you a brief summary. What we found is that cancer cells are innervated by nociceptor neuron, both in the mouse and in human. We found that cancer cells secrete SLIPy, activate nociceptor neuron, and that lead to the local release or the production of CGRP. So because there's a production of CGRP, and because we know that CGRP is a very strong immunomodulatory neuropeptide, we ask ourselves, is CGRP able to increase the expression of immune checkpoint receptor on CD8 T cell? So if we go on slide 23, we test this in various ways. For sake of time, I'm only showing one of the experiment. But basically what we did is we use a naive mice, we harvest the splenocyte, the naive T cell, we culture those T cell under TC1 inflammatory conditions. So we make those T cells cytotoxic. And basically we expose those T cell to CGRP. I think it was twice over the course of 4 days. And then we looked using flow cytometry at the expression of immune checkpoint receptor. And what we found is that in RAMP1 wild-type CD8 T cell, when they are exposed to CGRP, they overexpress or they co-express PD-1, LAG3, TIM3, which are 3 immune checkpoint receptor. We found that this response was absent in RAMP1 CD8 T cell. And then we looked at the expression of cytotoxic molecule. Here we see on the right panel is interferon gamma. We found that CGRP actually decreased the expression of cytotoxic molecule from CD8 T cell. And again, we found that in RAMP1 knockout CD8 T cell, this effect is absent. I'm not sure I mentioned it, but RAMP1 being the receptor of CGRP. So basically, culture T cells stimulate them with CGRP. They seems to be exhausted because they co-express PD-1, LAG3, and TIM3, and they lose the expression of interferon gamma. So if we go on slide 24, so basically one of the question is, whether the co-expression of these immune checkpoint receptor, right, it suggests that those T cells are actually exhausted. But the real test to see if those T cells are exhausted is to see, are they more or less able to eliminate cancer cell? So what we did is we cultured B16F10 ova cancer in the presence of an OT1 CD8 T cell. OT1 CD8 T cell are basically T cells that are engineered to specifically eliminate ova expressing cell. And so we culture those cancer cells with the ova specific CD8 T cell, and we found in the white that after 24 hours there's about 15 to 20% of the cancer cells that are being eliminated by the CD8 T cell. But when you stimulate the co-culture with CGRP, we found that there's a significant decrease in the elimination of the cancer cell by the T cell. So basically, taken together, if we go on slide 25, what we found is that CGRP is able to increase immune checkpoint receptor expression on T cell and that decrease their cytotoxic potential. We found that because they're less able to eliminate the cancer cell. So finally, our third and final question, we ask ourselves, is the presence or absence of the nociceptor would modulate cancer growth in vivo? So then if we go on slide 26, we first ask ourselves, and that was a cool question to ask, is pain actually preceding immune cell exhaustion in the tumor, right? Can we found that there's thermal hypersensitivity before the development of the T cell exhaustion? So this was a quite long experiment. What we did is we injected the cancer cell in the hind paw of the mouse, about, I think it was 120 mouse. And we followed the development of thermal pain hypersensitivity, we followed tumor growth, and we followed the development of immune cell exhaustion inside of the tumor. And we sacked the mice pretty much, I think it was every 3 days, and we looked at those different parameters. And what we found is that there's the presence of thermal hypersensitivity actually start on day 7, after tumor inoculation, while immune cell exhaustion actually start, I think it was day 11 or day 12. So these data actually showed or suggest that T cell exhaustion is actually coming after the development of thermal hypersensitivity. Then on the right-hand side of the graphic, we ask ourselves, and this was done after day 22, so with a big tumor, we ask ourselves, is the level of the thermal hypersensitivity can correlate with the T cell exhaustion inside of the tumor. And what we found is there's actually a pretty nice correlation. I mean, it's not super strong, but 0.55. That what we found is that there's a nice correlation between the development of thermal pain sensitivity and CD8 T cell exhaustion. So basically, high thermal pain, high T cell exhaustion, low thermal pain, low T cell exhaustion. So then if we go on slide 27, what we did is we decided to use, we decided actually to look at whether the presence or absence of the nociceptor would impact tumor growth, right? So we use a nociceptor neuron ablated mice, which is called TRPV1 dipterotoxin, and we use the littermate control mice, TRPV1 wild type dipterotoxin. And then we inoculate the tumor in the flank and we looked at tumor growth. And what we found is that in absence of nociceptor neuron, there's smaller tumor growth, and that on the right-hand side, we found that those mice actually survive longer. Then if we go on slide 28, what we did is we harvested the tumor and we looked at the immune cell inside of the tumor. And what we found is that in absence of nociceptor neuron, so in green, the CD8 T cell are more cytotoxic, so they express more interferon gamma. And on the right-hand side, what we found is that those T cells are also less exhausted. So there's less co-expression of PD-1, LAG3, and TIM3. Then if we go on slide 29, we ask ourselves, is this difference in tumor growth actually depend on the immune system? So to answer this question, we use two other type of melanoma. A melanoma that is considered to be immunogenic, like the B16F10, basically. So meaning that when you inoculate the tumor, it's known that the immune cells come inside of the tumor and will fight the cancer cells. So it's called Yummer 1.7. And we also use another type of melanoma, which is considered non-immunogenic, meaning when you inoculate the tumor, there's not really immune cells that come to fight the tumor. So in the immunogenic tumor, we found that ablating nociceptor neuron also decreased tumor growth. However, or in contrast, we found that in the non-immunogenic tumor, the presence or absence of the neuron did not impact tumor growth. So on slide 30, now we know that the difference in tumor growth is actually dependent on the tumor being immunogenic, right? So there's an immune response that's created against the tumor. So we ask ourselves, is the defect in tumor growth in absence of neuron depends on the presence of T cell? So to do this, we systemically depleted T cell using anti-CD3 and anti-CD8. And what we found is that when you deplete either CD3 or CD8 T cell, there's no difference in tumor growth, whether you have neuron or not. So these data shows that nociceptor neuron probably control the function of T cell against the tumor, right? So on slide 31, we actually went ahead and tested this possibility. What we use is a RAG1 knockout mice, so some mouse that do not have B or T cell. We inoculated this mouse with the cancer cell. We let the tumor grow to about 500 millimeter cube. Then in one group of mice, we ablated nociceptor using RTX, and then we give these mice naive OT1 CD8 T cell. And what we found is that in absence of nociceptor neuron, the OT1 CD8 T cell really decrease tumor growth much more strongly than in the mouse that has intact nociceptor neuron. And in these mice, when you harvest the T cell and you look at their exhaustion, we found again that when you ablate nociceptor neuron, the OT1 CD8 T cell were less exhausted. Then if we go on slide 33, basically, so we can skip 32, we can go directly on 33. So far our data have shown that if you ablate nociceptor neuron, you know, either using a genetic approach or using pharmacology, we see smaller tumor growth. Then we ask ourselves, what would happen if we activate nociceptor neuron? So what happened in the gain of function experiment? So we decided to use optogenetic to activate nociceptor neuron and see if the activation of nociceptor neuron in the tumor would change the way the tumor actually grows. So in this case, we use an NEV 1.8 Cree channel rhodopsin mice, and we expose the mice or the tumor to a blue light laser once a day for about 20 minutes. And then we looked at tumor growth. If you go on slide 34, we found that in mice that were not exposed to the laser in white, there's a normal tumor growth. However, in the mice that were exposed or were activated with the light, we found higher tumor growth. On the right-hand side of the graph, after we sacrificed the mice, we on day 14, we actually looked whether the activation of nociceptor neuron could increase the local release of neuropeptide, and we found that yes, it is the case. When you activate nociceptor, when you increase the activation of nociceptor, we found increased CGRP level inside of the tumor. I don't want to take too much time, but I think I'll briefly show the data on slide 36. So then we ask ourselves, can we block those nociceptor neurons using pharmacology? To do this, we use a charged lidocaine derivative, which is called QX314. So basically, lidocaine is an highly lipophilic drug, right? It will block all the function of all type of neuron, right? It diffuses inside the lipid bilayer of all type of neuron. It binds and block the function of voltage-gated sodium channel and prevent the generation of action potential. QX314 is actually a positively charged version of this lidocaine, and this positive charge prevent the diffusion of QX314 inside of the nociceptors. So if we looked on slide 37, basically what we ask ourselves is there enough ongoing inflammation inside of the tumor to tonically activate TRP channels, to tonically activate those nociceptors? So the TRP channels would be open, and that would let QX314 enter the nociceptor and block their function. And what we found is when you administer QX, I think it was once a day in and around the tumor, we found that there's smaller tumor growth, and we found that the T cells were less exhausted and they were more cytotoxic. Basically, what does that show is that we can use this lidocaine derivative to block locally the function of nociceptor neuron, and that would prevent or safeguard T cell from undergoing exhaustion. Then we can go on slide 39. So basically what we found is when you ablate nociceptor neuron, right, we decrease tumor growth. So nociceptor neuron are the type of neuron that secrete neuropeptide, and they are the type of neuron that secrete CGRP. So then we ask ourselves, is the defect in tumor growth depend in the presence of or in the absence of CGRP? So what we did is we actually rescue CGRP level in the tumor once a day, and we looked at tumor growth. And what we found is in nociceptor ablated mice where we rescue CGRP level, there's no defect in tumor growth, and the T cell show no defect in CD8 T cell exhaustion. Sorry about that. So basically, the defect in tumor growth depends in the absence of CGRP in nociceptor ablated mice. If we go on slide 40, what we decided to do really to drill down on the effect of CGRP is we went ahead and blocked specifically using an antagonist the CGRP receptor RAMP1. And what we found is when you block RAMP1, I think this was done once every 2 days systemically, is when you block RAMP1, we decrease tumor growth, we decrease T cell exhaustion, and we safeguard CD8 T cell exhaustion. And on slide 41, that was really the key experiment of the article, and really kind of showing the relationship or the role of the CGRP RAMP1 axis in tumor growth, right? So what we did here is we use a RAG1 knockout mouse, so a mouse that has no B and T cell, and we actually injected or given or rescue, what we did is actually, we transplanted RAMP1 wild-type CD8 T cell and RAMP1 knockout CD8 T cell in the RAG1 knockout mice. We give the mice the tumor, and we looked at tumor growth. And what we found in comparison to the pink group, which received the RAMP1 CD8 T cell, we found that the mouse that received the RAMP1 knockout CD8 T cell show smaller tumor growth. In the right side of the graphic, what we did is we were able to track whether RAMP1 are wild type or RAMP1 are knockout CD8 T cell using CD45.1 and CD45.2. So in some of the mice, what we did is we actually co-injected both RAMP1 wild type and RAMP1 knockout CD8 T cell, right? And using 45.1, 45.2, we're able to parse those two types of T cell inside the same tumor, and we found that the mice that received the RAMP1 knockout CD8 T cell show lower CD8 T cell exhaustion. After this experiment, we purified those T cells using flow cytometry, and we RNA sequenced them. As this is what you see on the bottom of the slide, basically, is that the RAMP1 knockout CD8 T cell would actually show lower expression of diverse immune checkpoint markers. So again, both transcriptomically and using flow cytometry, the RAMP1 knockout CD8 T cells show less T cell exhaustion. So if we go on slide 42, we ask ourselves, is this only true in the mouse? Is this also true in human? So what we did is we used the Broad Institute single cell atlas, which allows you to mine, single cell RNA sequencing data sets. And what we did is we mined those single cell RNA sequencing data set from melanoma patient, and because it is single cell, we're able to parse the T cell in two populations. Basically, do they express RAMP1? Do they not express RAMP1? And what we found is that in melanoma patient, the T cell that do express RAMP1 overexpress a lot of immune checkpoint receptor, including PD1, TIM3, LAG3, and CTLA4, and in comparison to the RAMP1 negative T cell. So our finding seems to be true also in human. And then when you looked on the right-hand side of this graph, basically we use OncoLink, and that allows you to basically track the survival of patient, whether they express high or low level of different protein. And what we found is that in human that express high RAMP1, they seem to have lower survival than in patient that express low RAMP1 inside of their tumor. We can skip 43 and go directly to 44. And then we ask our final question, basically, again, using patient data. Is the presence or absence of RAMP1 on T cell would impact the patient response to immune checkpoint blocker? Again, as I mentioned, we use those single cell data. We parse the T cell from the different patient, whether they are immune checkpoint resistant in red or they are immune checkpoint blocker responsive. And what we found is that in immune checkpoint responsive patient T cell, they express lower RAMP1 level than the immune checkpoint resistant patient. And then I think we looked at about 30-something different markers, and we found that RAMP1, the difference in RAMP1 expression between the resistant and the responsive T cell was actually ranking third in all the 30 different markers. So it seems that RAMP1 expression or higher RAMP1 expression on patient T cell could be a cool marker, an interesting marker to look at or to indicate that those patients would be likely unresponsive to those immune checkpoint blocker. So I'll conclude with slide 45, just to resume what we found. Basically in the skin, normal skin, you see that there's presence of diverse type of neuron, including sensory neuron. We found the presence of T cells that are likely not exhausted. When there's the development of melanoma, we see that there's increased tumor innervation and there's increased T cell exhaustion. We discovered that melanoma via the secretion of sleepy would sensitize the receptor neuron to locally release CGRP. CGRP would act on a RAMP1 receptor on the T cell, and that will lead to T cell exhaustion. And what we found is that if you block basically nociceptor neurons function using QX314 or using Botox, or if you block RAMP1 using BIBN, we're able to prevent T cell exhaustion. So we're able to basically safeguard the function of the cytotoxic T cell against the tumor, and that leads to basically a smaller tumor growth. And then if we go on slide 46, I'm just briefly, after our paper got published, or pretty much at the same time our paper got published, there was 3 other papers that got published in another journal. And just briefly mentioning that our findings seems to have been replicated by other investigators. Here we see on slide 46, there was a nice cell metabolism paper that found that when you ablate nociceptor neuron, they see also that there's smaller tumor growth, so smaller B16F10 growth inside of these mice. On slide 47, there was a paper by Nicole Sheff that actually found that when you block or when you knock out CGRP, she discovered that in head and neck cancer, there's smaller tumor growth and higher activity of the immune cells against the tumor. And finally, on slide 48, there was a very recent Nature Con paper that actually found that another neuropeptide, not CGRP in this case, but VIP, could lead to T cell exhaustion in the context of pancreatic cancer. So on the last slide, basically slide 49, this is the team. It's people that did the work. So all my thanks to them. They've been fantastic. And yeah, so thank you guys. And take any question if there's anything unclear.
Well, thank you so much for this amazing presentation and for guiding us through this really interesting and a lot of work. I think it was a lot of work, and but it's really you answered all the questions like it was like a crime,
Okay,
a
that's
puzzle.
good.
So that's wonderful, and it's really interesting how you dissected out that the pain receptor or the function of pain receptor, usually traditionally we thought about it will warn us that something is wrong, but it's actually contributing to something going more wrong.
Yes,
So
yes. Yeah,
that's,
yeah, yeah,
that
exactly.
was really surprising to me. And I don't know if that's something you would think about or if it would maybe not be possible to do, but do you think that there's already a pre-existing different, I don't know, gene expression or so, or usage of the gene before patients have cancer in
Right.
in their neurons?
Yeah, that's a great question. So that's something we're trying to look into whether say patient with higher, I don't know, let's say neuropathic pain, right? So some patient that would already be in pain, is there higher prevalence of cancer, or are these patients doing worse? And so on. Well, unfortunately, we don't have the answer to that yet, but we're really trying to leverage bioinformatic and the work of others to try to link the two, right? So patients that already have chronic pain, whether they have more or less tumor, are they doing more or less worse? Unfortunately, no data on this yet, but I think I I'm confident we'll find something interesting. And I'm saying that because you know how chemotherapy, right? It induces chemotherapy, CIPN, chemotherapy induce peripheral neuropathy. And so one of the question is that is that CIPN, or is the effect of chemo directly on the receptor neuron can negatively affect responses to chemo after, right? And we started to try this in the mouse, and it seems that if you ablate the neurons, the mice are more responsive to the chemo, and of course they don't develop pain because we ablate nociceptor neuron. So it seems to be like kind of um, two bird one stone situation where blocking neurons can increase responsiveness to the chemo and also prevent development of pain. And so let's see what happens in larger population of patient, right? Whether chronic pain can change development of malignancy, but at least for the chemo bit, which is, we know it's painful, it seems to be it seems that neurons are actually um, are bad, basically, for responsiveness to chemo.
Yeah, I was thinking because I worked with autism like
Mm-hmm.
mice and for a while, and they have also a difference where also patients with autism have a different perception of pain.
Right.
Like some have more, some have less, and there's a confusing data out there and publications that either there's autism with less risk of having cancer, like some types, and other types with having more, like a higher risk of cancer under age of 30. So I was
Okay.
thinking maybe if somebody would look into if that's correlated with their pain perception, it would be really interesting, um, data set to start with, but I'm not sure if it will lead to something.
Yeah, this is a cool idea. Yeah, I did not really think about looking at autism in that subset of patient, but that's a cool, yeah, that's a cool idea. I should, or we, or hi, or us, anyone, but we should look into this. Yeah, that's a cool idea. So yeah, thanks for pointing this out. That would be a cool thing to look at.
Well, thank you. And Serena, Dr. Shah, Denise, welcome to the stage. Did you have questions? Please unmute yourselves and just go ahead.
Yeah, so thank you so much for the talk. It's fascinating. It's a little outside my area, so
Thank you.
but I got the the question about so the cancer cells. You found the signaling was slippy, but it was sort of a follow-on question to Katarina's first. Does that actually alter the expression profile of the neurons? And into as a it's, I mean, it's fascinating to just think that the cancer cells have jacked the neurons into tricking the immune system into exhausting early as its own defense. But that the whole and your work about exposing the pathways is fantastic. But I guess that's an open question, as you were alluding to, whether the actual expression profile of the neurons are altered by the cancer cells, or if this is if they're just the presence of those neurons just exposes that vulnerability.
Yeah. So well, I guess so. So okay. I okay. So thanks for the question, and thanks for allowing me to rephrase, I guess, my answer. So yes. So when we co-culture, let's say, cancer cell with neuron, and then we RNA-seq these neuron, we do find that there's about 1,000 genes, I think, that are differentially expressed in these neurons. And then when we, let's say, give the cancer to the mouse, and then we use a retrograde tracer, right? And then we trace back these neurons that are innervating the tumor, and then we do single cell RNA-seq in these neurons. We also find that there's about, I think, 500 to 1,000 genes being differentially expressed in these tumor innervating neuron. And actually, this is the follow-up work that we're working on right now. We coined these neuron being tumor associated neuron, and they actually have a super cool expression profile where they're actually completely different than naive neuron, and they overexpress a bunch of different neuropeptide, of course, including CGRP, but a bunch of other that would or are actually fascinating to study. And so now we're trying to. That's the, I guess, problem with transcriptomic, right? Is to pick the right target after. There's many interesting little pathway that we're looking in these tumor-associated neurons. So basically, the short answer is yes, transcriptomic of the neuron are changed both in vitro and in vivo. So the cancer do change the transcriptome of the neurons.
Fascinating. And I suppose is it more is it generally appreciated, or is this a very different paradigm that the use of neurons in this the progression of cancer, because the cancer cells are jacking them and
Right.
reducing the immune system?
So I guess yeah, no, it's well, it's fairly new. So there was the work of Dr. Frenette, 2013, 2017, and from there there's a few super interesting paper that have been published looking at the role of neuron in different type of cancer, prostate, ovarian, um, head and neck melanoma. So I think it's definitely getting traction. Glioblastoma, of course. So there's a couple of fascinating paper that have been published by various investigators looking at this. But yeah, I think it's definitely getting traction, right? So over the past few years, there's more and more people investigating that, and you see, and this is really cool for us. We see in uh, in cancer, let's say a specialized conference. There's now a little cancer neuroscience bit included, part of these conferences. The same thing for like Society for Neuroscience and FENS. There's now like little bits focusing on cancer neuroscience. So I think it's definitely gaining traction, and hopefully that could lead to fun discovery for the patients.
That's amazing. Okay, I'll pass the mic. Thank you so much.
Thank you so much. Thank you.
Yeah, thank you so much, Sebastian, for your wonderful talk. And my question from you, by considering the CGRP that you just mentioned in your talk, and I was just wondering, because we are talking about the angiogenesis, and we know about all of those cofactors that they impact to the microenvironment, especially for the, angiogenesis. What you see the role of the, for example, mast cells, and did you work on a APC and focusing on a immunotherapy based on activation of the APC or optimization of the APC or not?
Yeah, thank you for the question. Yes, so I have a PhD student right now looking at APCs in this very context. So whether the presence or absence of tumor would modulate the function of CDC1, so that subtype of dendritic cells, right? And so far, it seems to show that when we modulate the function of nociceptor, CDC1 seems to be more able to uptake tumor antigen, and that actually increase their trafficking from the tumor to the lymph node. For the mast cell specifically, we did the single cell of the tumor presence or absence of neurons, right? We see that there's changes to the mast cell, but the changes are limited. So the transcriptome of the mast cell do not seem that affected. So for now, we're really focusing on the APCs and on NK cell as well. So we see NK cell, there's big changes. That being said, crosstalk between nociceptor and mast cell have been studied in the skin. Nicola Godenzio has a very nice paper in the context of dermatitis, which shows like very, very strong crosstalk between skin, nociceptor neuron, and mast cells in the context of allergy.
Yeah, especially when we are thinking about the lymphocyte and allergy pattern,
For sure.
and when you know about the pro-angiogenic factors that you're just talking about. And did you just find out any matrix metalloproteins presence in the microenvironment?
Right. I don't think I actually never. I don't think we looked at it by ELISA's. Yeah, this is
And what
something we
about
did.
cationization? That's why I ask.
Right? No. Yeah. No, I understand. Yeah, unfortunately, no. Good question. We never looked. Right? Yeah. So I guess that's the fun part with the project where we could look at cancer. There's so much to look. Just angiogenesis per se, we never looked. Um, Cancer-associated fibroblasts, and as you mentioned, MMPs, and so there's I guess many open questions for everyone to look at.
For sure. Thank you so much. I'm passing the mic to Dennis.
Thank you.
Thank you, everybody. Thank you, Sebastian, for this really interesting presentation. I was very, I really like the visuals I'm a visual person. I'm sure I'm not the only one.
Okay. Okay.
You had mentioned Botox during sort of the end of the talk, and I wasn't really exactly sure
Yeah.
what you meant. Could you recap that part?
Sure. Absolutely. And then I actually did not show the data, but but it, it was part of the schematic. But so it's in the article. We also try to use Botox. So what we did is we locally injected Botox in the skin. Then we give the mice the tumor. I think it was, we injected Botox on day 1 and day 3, and then 2 days later we give them the tumor. And what we found is when you block function of neurons, but we know that Botox is not only nociceptor neuron but all types of neurons. We found that there was similar finding, right? Smaller tumor growth and less immune cell exhaustion. So from the Botox data, then we wanted to only look at nociceptor, and this is when we use the charge lidocaine derivative. So I did not, unfortunately, did not show the Botox data today, but yeah.
That's okay. I just wonder if Botox proponents will be like, "See, this is amazing. It helps your
I know, I
your
know.
T cells."
I know, I know, no, yeah, yeah, yeah. And I have there's another investigator which I did my postdoc with, Isaac Chu at Harvard, which found that when you give Botox in the context of bacterial infection, that was also basically improving or decreasing bacterial growth. So again, yeah, you're increasing the function of the immune system against the bacteria. So Botox seems to have many, maybe many benefits.
Yeah, it's very interesting, right? Like you're paralyzing the muscle, but then you're increasing the cellular activity underneath it.
Exactly, exactly, exactly.
That's not exactly that's definitely something I would not have expected.
Right. Yeah, 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 for sure. I mean, yeah, I mean, Botox, yeah, in different contexts, actually, it could be quite interesting to look at.
Oh,
So,
oh boy.
yeah, exactly, exactly.
So
Yep,
I was
yep.
also curious. So how long did this take? Because there was so much information. It was so detailed, and you have ran like so many controls. Like, how long did this take? What was the staff? What was the budget? Like, what's
So
that?
it took 8 years.
Oh
Um,
wow.
yeah, yeah. So 6 year from the first submission, and then 2 years of revision, and the staff. I mean, yeah, many people, many other labs, right? It's not just our lab. So many collaborator, and the budget. I honestly, I don't want to make the total because I think I would be sad. Um,
Understandable.
yeah, yeah. So it was yeah, definitely. So high, I would say, too high, but yes, yeah. So maybe it's probably my first big grand pass on that project. So definitely close to $1 million, I think.
Well, congratulations on that part. I have two last questions.
Yes.
How likely is it that the dynamics that you found in mice would be identical in humans, or substantially similar?
No, that's a fantastic question. So when we looked at those database, based on the database, it seems to potentially be true as well in human. Um, and I think the next step now, we're trying to leverage collaboration with companies where, all these companies that have the anti-CGRP for migraine, and we're trying to see. And the problem is those drugs are in the market for 2, 3, 4 years, but we're trying to see whether they have any sorts of data that would show that people that take the anti-CGRP drug for migraine are these patients actually have less malignancy, or if some of these patients also had malignancy and took the migraine drug, are those patients actually doing better, right? So we're chatting with a couple of companies to see if we're able to, if they are able to basically mine their data and see if there's some link. So hopefully, hopefully we'll both show that, patients that take the migraine drugs or the anti-CGRP drug, maybe if they would have malignancy, maybe they are doing better than not their peers, but their well, like control or non-migraine drug treated patient, right? So,
Mm-hmm. The
but yeah,
control.
I mean, the data set seems to support the idea, but I guess we'll need to see. One of the other aspect that would be interesting to do is to try those PDX mice, where you take tumor from patient and put it in humanized mice, right?
Mm-hmm.
And see if we're able to replicate our data. So that's the next that's our next big step.
Right on. Well, definitely good luck to you on that last step.
Thank
And
you.
so, because we were talking about, the main part of this was about immune system and measuring those markers, I was just curious if you had any speculation about what these findings might mean for COVID patients, like chronic COVID patients primarily, not so much the acute ones.
Yeah. So half of my lab is actually working on lung inflammation, and we try to block a lung neuron from these ACE2 mice, and then we give them the COVID. And unfortunately, actually, we found that when you block lung nociceptor, the mice were doing worse.
Hmm.
So yeah, but as whether or not it would impact, let's say, long COVID, this remain. I so there was a few article that looked at the possibility using, let's say, bioinformatic, and it seems that it would be potentially interesting. But the work we did in the mouse, at least in our end, right, it did not seems to be um, supporting the idea that blocking nociceptor would improve the immune system response against COVID, unfortunately.
Well, I mean, it would be great if it was better news, but at least we have the data, right?
Right. So we're trying to put this together. We're trying to publish those negative data. Let's see how that goes. Right.
For sure. All right. Thank you so much for
Thank you. Thank
everything,
you.
your time. I guess I'll pass the mic back to Katerina.
Yeah, thank you so much. Do you still have time for two more questions
Yes.
from two people? Thank you. So, Brooks, welcome. Did you want to ask a question?
Oh yeah, it's very interesting. So, Sebastian, I don't know anything about you because you don't have a profile created. So, I'm just thinking that it's new. It makes
Yeah.
it easier once you do create that profile for people to come up on stage and not have to ask you, oh, okay, what's your background? Where'd you study? What'd you do? Are you
Sure.
an experimental scientist?
Well, if you,
Uh,
yeah, I mean, we did the introduction with like where Dr. Talbot went to school and everything. If you
Yeah, but this is
scroll up in the
Clubhouse.
chat, yeah, but we have uh, every time we
This
invite
is
actual
Science Society.
scientists. So we invite actual scientists. If you want to scroll up in the
I'm not a
chat,
scientist. I'm a
okay,
physicist, and I work in the experimental field. So I was just asking him if he was just like me, just from a
uh,
different platform or discipline.
yeah. So, well, which is one of the cool part today. I was not really familiar with Clubhouse, so this is great. It's fun first
Well,
experience. So I'll definitely add the profile. I was actually so I downloaded the app and whatnot today. But so yeah, to quickly answer your question, I'm a PhD. I'm trained as pharmacology and neuroscience. So not physician, but yeah.
do you work in the experimental field?
Yep.
Good, good. Yeah, me too. I'm an experimental physicist. And before Clubhouse became what it became, it actually was closed. And it used to be just nothing but intellectual conversations. It was professors from all different
Ah,
disciplines and universities. It was nice. Now it's about like just a lot of people. So welcome. And I'm sorry it took so long for you to get here.
well,
Um, so can I ask you what X is your scope that you use?
can you repeat that? Sorry.
So the magnification of the scopes that you use?
For like
Data
the
collection?
Well, Brooks, if you want to look up the paper, as I said in the chat, I shared the lab website of Dr. Talbot and also the paper, and then the method section, you would find that answer. But if you have a question about the talk, so just to let you know, here in Science Society, we always invite the scientists that actually do the research of published peer-reviewed papers to present their work. So if you come here, you can, verify and expect that it's the actual scientists that did the work that are here presenting. And if you want to look that up, it will be really easy to find it in the methods.
Katerina, are you a member of the Quantum Photonics Club?
No, I'm not.
Okay, good.
yeah, as
Okay, so
far
the
as
question,
the
so
yeah
I studied
yeah, go ahead.
neuroscience
Sorry.
deeply, and so you're saying that there's a certain neuron that detects cancer, and it's something that must have just been evolved just recently to detect something that's problematic. It's a problem for our society, but it's actually. Have you ever heard of what was it? It was Helicobacter pylori, and how
Mhm.
it causes cancer to subside because of temperature and difference. Have you tried using that in a petri?
No. Okay, so H. pylori and temperature. No, I was not aware of that actually. I'll
Yeah,
need to.
it causes cancers and tumors in soft tissue to dissolve and go into remission.
Huh. Well, that's super interesting. Yeah, I'll definitely need to look into this. I'm fortunately not familiar with this work, but
Do you see why all our paths are supposed to cross at the right time? That's what Clubhouse does.
yeah. No, for sure. Yeah. I'll look into this. Thanks for pointing this out.
Of course. Thanks for taking the time to listen to me. Enjoy the
Thank
day.
you. Thank you. Have a good one.
Okay, thank you so much. But in general, Helicobacter pylori induces gastric cancer, and it's a high risk factor, and so on.
Right.
Yeah, we have to confirm that. So well, Sebastian, thank you so much. Let me just check really quick the chat again if somebody ask more questions, but so far I think no. So this was such an interesting discussion, and your work is really amazing. I'm really curious to follow what your lab does in the
Thank you.
future. So I'm curious to learn more, and maybe one day in a year or so you'll come back and share some more with us. It's really interesting,
Would
and
love
I wish
would
you
love
a lot of funding
Yeah,
and
thank
a lot
you.
of luck.
Thank you. Thanks to everyone being here and maybe just close. If there's we're looking for grad and postdocs. So if anyone's interested or if you want to share in your network, um, you know, feel free to reach out. We have position opens. So yeah,
Yeah, you send me if you want to send me like a job post I can share.
definitely. I'm
Also,
working on the job post, but I'll definitely send it to you, Katerina. If you
yeah,
can post it on Clubhouse, that would be great.
yeah, here and on Twitter, and I'll share it my community, like my PhD, like it was GABA. It's from Portugal. Like it used to be an
Yeah,
organization, but there are people maybe also looking for jobs.
for
And
sure.
yeah, great. I wish
Fantastic.
you that you get amazing, great people. can, it can sometimes go both sides, but
Oh
no,
yeah, yeah, for
usually
sure, for sure, for sure.
usually it should be good. But okay,
All
then
right,
good
guys.
luck.
Well,
Have a
yeah,
great
yeah.
day. Thank you so much again. This was really wonderful.
Thank you, guys. Cheers. Bye bye.
Thank you.
Okay, and if you like discussions like this, follow the club, and we'll have more presentations in the future. Check out the schedule. We'll have next Dr. Ningebor talking about high resolution imaging of gene folding, like the new ways of how the genes are folding and how we can image that in more detail. I think that's really interesting work. So I hope to hear you all back soon. And thank you, Sebastian. Thank you, everyone, for coming, asking questions. This makes the discussion so much more interesting if more people participate. So thank you so much. Okay,
Absolutely. Cheers.
cheers. I close the room in 3, 2, 1. Bye, everyone. Thank
Thanks.
you.
All right.
Bye.
Bye bye.